oh.
So we have watched carefully the lecture I uploaded. There were three important concepts there. And the three important concepts which you should be learning as part of this particular lecture are, so we talked about their DNA replication. We talked about DNA as molecule for hereditary molecule. And we also talked about DNA damage repair. Now, when we talk about DNA, we have three words: DNA, gene, and chromosomes. As freshmen, you should know that these are all three are linked. DNA, as you all know, is made up of 4 nucleotides. It's a double helical structure, and DNA, 146 base pairs of DNA together with 4 different histone proteins. And these histone proteins are H2A, H2B, H3, H4. 2 molecules of each of this histone, along with 146 base pairs of DNA, it constitutes a structure called nucleosome. Nucleosome. And nucleosome is the fundamental particle of a chromosome. So normally, you know chromosomes in books. You must have seen chromosomes like this. Like this, okay? This is not a normal chromosome. This is actually a mitotic chromosome. This is a chromosome in a cell which is ready for cell division. Normally, a chromosome is like this. And if I try to draw DNA here, you will have this double helical structure there. It's not a linear molecule like this. It's a double helical structure. The nucleosomes, many nucleosomes, they are supercoiled, okay? And so you see now the relationship between DNA and chromosome, that chromosome is made up of DNA wrapped on nucleosomal particles, and each chromosome is like millions of nucleosomes. Each chromosome is millions of nucleosomes, maybe, based on what is the total size of the genome in an organism. Now, what is a gene then? So we did talk about DNA, we know about chromosome. Gene is basically a specific length of DNA which contains very specialized sequence, which we call promoter. Please mute your mics. And it includes specialized nucleotide sequence, which we call coding sequence. And then there is specialized sequence of DNA, which is called termination sequence, where the process of transcription gets terminated. We will talk about detail of gene in the next lecture, but for today, what you should know is that a tiny part, a very small part of a chromosome is a gene. And on a single chromosome, there could be hundreds and thousands of genes. Now, we have to focus on the concept which was DNA replication. Why DNA replication is required? We all start our life from single cell. This single cell is formed after fusion of sperm and egg cell. And that is the day one of your life when we call this cell 2N, the diploid cell, because it contains. Are there questions? Is there a question?
Sir, do we wait for like a certain part, or can we ask a question related to the lecture at any time?
You can ask me question anytime.
Sir, I have a question about the genome.
Hmm.
So when we say, like, further in the lecture, in your recorded lecture, you said within a cell, all the present genes are the genome of that cell. So when is it the same for when we say like an environment? Within an environment, all the present genes in all organisms, is that the genome for that environment? Is that correct to say?
Yeah, so I'm just, so your question is right at the time. I'm just coming there. So when
Okay.
a human sperm, which carries 23 chromosomes, and egg contains 23 chromosomes, when they fuse, so this cell is called 2N, which is basically 2N means diploid. Okay. And the sperm and egg cells, which contain half the number of chromosomes, they are called haploids. Okay. When they fuse, we restore 46 chromosomes on our day one. Each one of us, when we were in mother's womb, on day one, when we were just starting our life, we had 46 chromosomes. So these 46 chromosomes, they constitute our genome of a single cell. Now, this cell needs to divide into two cells. That's how life starts. Each of this cell divides again into two cells. So we keep dividing and we are going through mitosis, okay? And we look like, we will see in a couple of lectures down this module, we become a clump of cells, okay? Couple of hundred cells we become, like a small ball of cells. Each cell here contains 46 chromosomes. Because this cell is passing on its DNA after the process of DNA replication. So now you should know that when one cell divides into two, the genome has to be duplicated. And what is genome? 46 chromosomes. And what is chromosome? It's histones plus DNA. And what is the size of our genome in a single cell? In a single cell, we are 6 billion base pairs. And in a haploid cell where we have sperm or oocyte, we are 3 billion base pairs. When we say a single cell goes to two cells, it means all these 6 billion base pairs, they need to replicate. We need to have 12 billion base pairs before a single cell will pass on 6 billion to each, which means 46 chromosomes to each cell after mitosis. Did I answer your question?
Yes, sir.
If there's any question, so the purpose of sharing the lecture ahead of time is that in this lecture, you can stop me anytime and ask questions. So if there are no
Sir, I
questions,
have a question.
she's an
So like you said कि जो है like जो हमारा जो sperm or like the egg cell, it has 23 chromosomes, right?
yeah.
So when they combine, they make a 40 chromosomal zygote, right? Like a haploid one, right?
Zygote is diploid, 2N.
Yeah, like but the sperm and the eggs are haploid, right?
Exactly. So
Okay. So
what's the
you
question?
said that the haploid ones have 3 billion base pairs, right?
Yeah.
Okay, so these combine to make the final like diploid, which has in total 6 billion base pairs, right? Because I get confused about these numbers.
Yeah. 23 chromosomes in sperm, 23 chromosomes in oocyte. When they fuse together, they make 46 chromosomes, which is a diploid. And the total number of base pairs on 46 chromosomes is 6 billion. And half of that, 1N, is 3 billion. Clear?
Yes, sir,
Sir,
it's clear. Thank you.
sir, I wanted
So,
to ask a question.
yeah.
Sir, in the beginning of your recorded lecture, you were mentioning of the four histone proteins and how they are formed, like placed in the form of pulley, and DNA is coiled around them.
Yeah.
Can you please explain that part again? I had like certain confusion about it. And the another one was the terminus and the starting vola section.
So, histone proteins, four different proteins, which are named histone H3, they are very small size proteins, 11 kilodalton, 17 kilodalton, H2A, H2B, and H4. H3 and H4, they make a tetramer, which means two molecules of each, H3, and two molecules of H4. Okay? And H2A and H2B make, so two dimers of H2A, H2B. They together, when they, this tetramer plus two dimers each of H2A, H2B, 8 molecules come together here, and they are wrapped around, so 146 base pairs of DNA is wrapped around them. This is called nucleosome. This is fundamental particle of chromosome. It's explained in the lecture. Is it clear now?
Yes, sir, it's clear. So can you please go
These
over the
proteins
part
are
of the
highly basic. The nature of
okay.
these proteins, they are highly basic. They are rich in basic amino acids like uh, arginines, lysines, etc. G, what else you wanted to ask?
Sir, there was a point where you were explaining K protein like starting point, but then comes a gene, and then there's this terminus. Can
Yeah.
you get that one?
So what I explained you, that the DNA synthesis takes place in 5' to 3' orientation. And there we did talk about why it is 5 to 3, because the phosphate group on the uh, fifth carbon is free, and always chain elongation takes place. The phosphodiester bond takes place at the 3' free hydroxyl group. And this is the way how chain elongation in DNA takes place. In case of protein, protein has amino and then the carboxyl group. Protein is made up of what? Amino acids. Amino acids are the building blocks of proteins. So you have a single amino acid has amino group and carboxyl group, and then there are some other residues. If new amino acid peptide bond is going to be formed, it's, so this side where the amino group is, this is called the N terminus, and this is the C terminus of protein. The new peptide bond will be formed here, where this will be released and the peptide bond is formed. So you have amino acid elongation go like this, CON, and then the last amino acid will have free carboxyl group. So this will be the amino group of the protein, and this will be the carboxyl end of the protein, C term of the protein. Okay, is it clear, Age?
Okay. Yes, sir.
Okay.
Thus, yeah, yes, sir. Thank you.
Okay, now let's come to the point, the concept I want you to understand is the DNA replication in the context of mitosis and cell cycle. You have already covered the cell cycle, I know. Okay. Now, when I say that a cell, let's say a human cell, has to divide into two cells, the mother cell has 46 chromosomes, daughter must have 46 chromosomes, otherwise they are going to be dead or abnormal cells. Now, these 46 chromosomes, this is a diploid cell. How they are diploid? Because we learned in the lecture that each chromosome exists in pairs. Let's say this is chromosome 1. Identical chromosomes, let's say this is chromosome 1 of human. We call them homologs. Homolog chromosomes. Then chromosome 2 may look like this. Chromosome 3 may look like this. Chromosome 4 may look like this. And this way we have, let's say, 46 chromosomes. DNA present on these chromosomes constitutes 6 billion base pairs. Now, when a cell decides that I'm going to divide into two daughter cells, what it is going to tell itself that I need to first replicate all the DNA present on chromosomes. All 6 billion base pairs need to be copied, need to be replicated. Okay. You studied the cell cycle. Dr. Faisal has covered that you have the G1 of the cell cycle, S phase of the cell cycle, G2 of the cell cycle, and then finally this is the mitosis or cell division phase, where cell eventually divides into two, these two cells. Before this phase, when cell decides I'm going to divide into two cells, the first thing it does, it starts replicating its DNA, and that phase of the cell cycle is called synthesis phase, where DNA replication takes place. We have 46 chromosomes. How this replication is going to take place? So let's look at a linear DNA on a chromosome. I'm just trying to draw a small piece of DNA, just a small piece of DNA, which is part of a chromosome. This double-stranded DNA on a chromosome. Now, you should know that a human cell, from the time it decides that I'm going to divide at this particular point and enter into S phase, from here all the way to this time, it has only 18 to 24 hours. And this DNA replication phase is even part of this cell cycle. It means all those 6 billion base pairs are to be replicated, are to be copied within this S phase. No errors. Okay. So in eukaryotes, within DNA, we have specialized sequence, very, very specialized sequences, because our genome is huge, and we call these sequences origin of replication. This is a very specialized sequence, a very specialized code in the DNA. And as soon as cell decides, all the chromosomes, all 46 chromosomes, their origin is denatured. Denatured means DNA at that particular position becomes denatured. The hydrogen bonds are broken and DNA becomes separated, yet they remain part of the same chromosome. because we have to prepare a copy of this whole chromosomal DNA. And it has to happen on all 46 chromosomes. So who does this? The enzyme is called helicase. DNA helicase is the enzyme which is going to break the hydrogen, all these hydrogen bonds, and DNA becomes, so let's say this is the 5 prime, 3 prime, 5 prime, and 3 prime. We have multiple origins of replication because our genome size is huge. Unless we don't have multiple origins of replication, we won't be able to copy all the DNA on a chromosome. Now, let's move to the next slide. I just take one small chunk and zoom in, okay? DNA is unwound, single-stranded, like this is the origin of replication. This is still the double-stranded DNA. This is still the double-stranded DNA on both sides. This is 5 prime and this is 3 prime. 5 prime and 3 prime. Now, on this origin of replication, cell receives a signal that you have to copy, start copying this DNA, both the strands. Let's say the helicase, the enzyme, which is bound here, it's moving in this direction. It's zipping apart the DNA. It's breaking down, and this replication bubble is becoming bigger and bigger. But the one which is naked here, now this is the direction of the replication fork. In order to copy this, you need some primer sequence. You need to have something where to add some copy nucleotide. Let's say here we have A, T, C, A, G, A, C, A, T, just the DNA sequence. Now, if you opened up here, now here, I just extend the sequence further. At this position, and another enzyme comes in, which we call primase. Primase is the one which adds just a small RNA molecule, single-stranded RNA molecule. Okay. Now, let's say RNA molecule is added here, and its sequence is G, U, C, U, okay, like this. This is 3 to 5, and this is 5 to 3. Since this is moving in this direction, the enzyme which is going to synthesize now DNA. So primase added the primer, DNA polymerase. DNA polymerase is an enzyme, DNA polymerase 3, which synthesizes DNA by copying a DNA. This will recognize this free 3' hydroxyl group and add copy complementary nucleotide G, then sorry, A, T, etc. As the helicase keep moving in this direction, this DNA will be synthesized in this direction. So what we have 5 to 3, and this is a complementary strand. So we have 2 anti-parallel strands going on. However, the trick comes in if the replication fork is going in this direction. Now this DNA strand is 5 to 3. 5 to 3. Now we cannot, while going in this direction, so primase, the function of primase is, it is going to add 5 to 3 RNA primer, which is complementary to DNA sequence here. Now, what is going to happen? The DNA polymerase, recognizing this free hydroxyl group, will synthesize the complementary nucleotides and copy this DNA. However, now as the replication fork moves in this direction, You cannot extend DNA from here because DNA synthesis it cannot go like, this direction. So this strand is copied in bits and pieces. In this strand is replicated or copied discontinuously. Discontinuously. Another RNA primer will be added here, 5 to 3, and then DNA polymerase 3 will extend this. So these short fragments on discontinuous strand, we call them Okazaki fragments. But this strand is called continuous strand. Continuous strand means without any interruption, it is being copied, but this one is discontinuous because RNA primer, then as the DNA is becoming naked, you have, these short fragments are being formed. Yeah, there are questions there. Please go ahead.
Sir, so the strand at the bottom, why does it have the DNA, I mean, primer cube under it? Isn't that the leading strand?
No, primer दोनों पे बनेगा because DNA polymerase, this is a very important point, DNA polymerase cannot synthesize DNA from scratch. We call in biology, we call de novo. It cannot just come in and say, okay, I add thymine here. No, it cannot do this. It requires a free hydroxyl group. Did I answer your question?
Yeah, but like my question is that I get that 5' to 3' wale strand pe, it makes the fragments because it's adding the primer in like different areas, right?
Yeah.
But the strand that's 3' to 5', wasn't that like the continuous one?
This
So
one is
why is it
continuous. This
yeah,
one
the
is
one at the bottom?
continuous. Yeah.
Okay, so starting from there and it'll continue on. Okay,
It
okay.
will continue 5 to 3. Since this one is discontinuous, primer, then some pieces, then another primer, then 5 to 3 elongation, then another primer, 5 to 3 elongation. Clear?
Sir, just confirming, the primer is added on the part from where the replication starts, right?
Yeah, they start from there, from origin of replication, but they will be on the continuous strand, you only need one primer. But in case of Okazaki fragment in discontinuous strand, let's say the replication bubble, let me draw it like this now. Like this. Like this, okay? Now, let's say this is your continuous strand. Clear? As a replication fork keep going, there will be 5 to 3 primer and then replication. New primer, because it will take time, the discontinuous strand. Then again 5 to 3, then again replication, like this. Clear? So we call these short fragments the Okazaki fragments. Now,
सर जो the Okazaki fragments जो है वो
Yeah.
एक specific length के fragments है
yeah,
units हैं.
yeah, they are like, the primer is, the RNA primer is very few nucleotide. I think if I remember correctly, and I may be wrong, 6 to 8 nucleotides. And then after equal space or stretch, when the DNA is naked, a new RNA primer is added. So on equal spacing you will have, and then these are short fragments. When DNA polymerase will synthesize, it will hit the pre-existing primer, it will stop replication here. Clear?
All right.
Any other question? So now, what is
Sir,
the consequence? जी, इर्तजा?
जी, I'm still a bit confused as to, well, ये decide किस तरह होता है कि कौन सी DNA strand जो है हमारी continuous बनती है, कौन सी नहीं बनती? Is it the presence of the primer itself? As in वो randomly किसी भी DNA strand पर attach होके वो कैसे?
No. Look, I told you in the lecture, and again I'm repeating. So orientation of this DNA is 5 to 3.
जी, इतना तो पता है.
तो इसका कॉम्प्लीमेंट्री यही होगा ना,
3 प्राइम टू 5 प्राइम उसका जो कॉम्प्लीमेंट्री होगा, ठीक
इसका
है।
कॉम्प्लीमेंट्री क्या होगा?
3
इसे
टू
यूं लिख
5।
लेते हैं, लेट्स राइट इट लाइक दिस, ज्यादा सिंपल हो जाएगा। 5 प्राइम और 3 प्राइम ये वाली स्ट्रैंड है जो कॉपी हो रही है, ठीक है।
ओके।
तो इसका कॉम्प्लीमेंट्री क्या होगा?
5 और 3।
5 किधर होगा?
5 will be up next to the 3.
इधर होगा, ठीक है। तो इधर 3 होगा
जी,
ना, तो प्राइमर का
एग्जैक्टली।
3 इधर है। तो कौन डिसाइड कर रहा है कि प्राइमर डिसाइड कर रहा है या ये वाली स्ट्रैंड बता रही है कि कॉन्टिन्यूस कौन सी होगी और डिसकॉन्टिन्यूस कौन सी होगी? आप बताएं।
जो रेप्लिकेट हो रही है, दैट सो बेसिकली द स्ट्रैंड व्हिच इज रेप्लिकेटिंग वो डिसाइड करती है कि कौन सी कंटिन्यूस कौन सी डिसकंटिन्यूस बनेगी।
लेकिन
इफ
वो
आई
जो प्री एग्जिस्टिंग टेम्पलेट है उससे डिसाइड हो रहा है,
ओह
इसका एंटी
ओके,
पैरेलल ही होगा ना।
जी जी,
ठीक,
ओके,
क्लियर?
ऑलराइट, दैट मेक्स सेंस, थैंक यू सो मच सर।
तो लेट्स सी नाउ। अच्छा हुआ सही टाइम पे एतजा ने क्वेश्चन पूछ लिया, क्योंकि अब हम आ रहे हैं कि ये हमारा टेम्पलेट था, इसमें लैगिंग भी बन गई और इसको यूं कर लेते हैं, ठीक है। बाकी क्रोमोसोम भी रेप्लिकेट हो रहा है। यहां पे आपका कॉन्टिन्यूस चला गया, लेट्स से 3 5, ये 5 3, और ये आपका डिसकॉन्टिन्यूस जो बने डीएनए, इसे यूं कर लेते हैं ना, वेवी डिसकॉन्टिन्यूस को। नाउ व्हाट वी हैव हियर, वी हैव आरएनए मॉलिक्यूल उसके अंदर है, क्लियर। आरएनए प्राइमर है, इसमें भी आरएनए प्राइमर है, और यहां पे गैप्स भी हैं, देयर आर गैप्स एज वेल बिकॉज इट स्टॉप्ड हियर, देन इट स्टॉप्ड देयर बिफोर द आरएनए प्राइमर। व्हाट हैपेंस after DNA polymerase performs its function of elongating DNA, another polymerase comes in which is called DNA polymerase 1. Its function is it degrades the RNA primers and it adds DNA sequence instead of this, क्योंकि अब आपके पास क्या है, इधर 3 प्राइम हाइड्रोक्सिल ग्रुप है ना, तो वो आरएनए प्राइमर को डिग्रेड करके इधर एक्सटेंड कर लेता है, आरएनए प्राइमर को डिग्रेड करके इधर एक्सटेंड कर देता है, नंबर 1। नंबर 2, अभी भी गैप्स हैं, जब होता है तो देयर इज एन एंजाइम कॉल्ड लाइगेज। लाइगेज इज एन एंजाइम which performs function of glue या जो भी चीज है। ये जो फ्रेगमेंट्स बने हैं उनके अंदर जो लास्ट, लेट्स से from here to here is one complete fragment after degrading the RNA primer, and then the next one is here. There is still a phosphodiester bond missing here. So ligase makes that phosphodiester bond, and the end result is what? End result is we have now one parental strand, I write it down, parental strand, and one newly replicated DNA strand. And on the other side also, this side, we have one parental strand and the other is new strand. So we call this semi-conservative mode of DNA replication in that lecture, which means the parental strand remains there and a complete new strand is synthesized. After the DNA replication, now how the chromosomes look like? We said in the S phase of the cell cycle, complete chromosome is duplicated. So after DNA replication, what we have, chromosomes before S phase, they look like this. Let's say this is chromosome 1, दोनों homologs हैं. This is chromosome 2, दोनों homologs हैं. Each chromosome, this is independent chromosome, this is its homolog. Each chromosome is one complete molecule of DNA wrapped around on histone proteins, and then it makes such a structure. And its identical chromosome homolog is this one. Now, after DNA replication, this chromosome will look like this. Why it's looking like this, and this one will look like this. Now we call them, they are two homolog chromosomes, but we call them this and this. These are called sister chromatids, and these two are also sister chromatids. Now, what has happened as a result of DNA replication? All the DNA which was present on this chromosome, it's copied, and it's part of the, it's attached to same chromosome. All the DNA here in the mother cell is copied in the form of a sister chromatid. We can't see them during S phase. We can't see such structures. They are only visible when cell enters into mitosis in this phase. In mitosis, there is a phase of mitosis called metaphase. These crisscross or X-shaped chromosomes are only visible there. Now, what is happening? Let's write it down. G1 phase of the cell cycle, S phase. In S phase, we cannot visibly see, but this has happened. In the G2, every nucleotide, all 6 billion base pairs, they are confirmed that they are duplicated. DNA replication has taken place. And once the scanning or proofreading has taken place, मैं भी उस पे आता हूं proofreading क्या है, the cell enters into cell division mode or mitosis, and then within the cell division, one of the phase of mitosis is metaphase. ये पढ़ा है ना आप लोगों ने? And in metaphase, we can see these chromosomes visible in such a shape. And what happens, वो आपने spindle fibers cell division पढ़ी है कि नहीं आपने? Anyone saying yes, no?
Yes, sir.
Yeah.
Yes, sir.
Yes, sir.
ये homolog chromosomes, homologs यहाँ पे आ जाते हैं, align हो जाते हैं like this. मैं सिर्फ एक all 46 will align like this. And what happens? The sister chromatids are pulled apart. ये पूरे का पूरा sister chromatid इधर आ जाता है, और ये वाला sister chromatid इधर आ जाता है. इसका जो homolog का है, ये वाला इधर चला गया, ये इधर चला गया. तो सेल डिवीजन के बाद क्या हुआ एज ए रिजल्ट ऑफ डीएनए रेप्लिकेशन? ये रिस्टोर हो गया ना स्टेट. ये एक डॉटर सेल में दोनों होमोलॉग आ गए, ये एक डॉटर सेल में आ गए. इज इट क्लियर सो फार? इज देयर एनी क्वेश्चन? एनी क्वेश्चन?
Sir, I have a question.
जी प्लीज गो अहेड.
Sir, what's the function of the centromere in the sister chromatids? Is it the same as the origin of replication, वो जो DNA के बैक्टीरिया के DNA में होता है?
No. So centromere का फंक्शन ये होता है, the spindle fiber, the microtubules, these cytoskeletal proteins, ये thread like proteins होती हैं, they specifically bind to the DNA sequence which we call centromere. This is the sen sequence, centromere. Microtubule इनको bind करता है और इन पे physical force apply करती है, और यहाँ पे माइक्रोट्यूब्यूल पे पूरा एक प्रोटीन्स का कॉम्प्लेक्स होता है जिन्हें हम कहते हैं काइनेटोकोर कॉम्प्लेक्स. प्रोटीन्स होती हैं जो इस पे बाइंड हुई होती हैं, और वो माइक्रोट्यूब्यूल के साथ बाइंड करके ये स्ट्रक्चर है क्रोमोसोम का, ये वाला, इन दो सिस्टर क्रोमेटिड्स को सेपरेट कर देती. क्लियर?
Yes, sir. Thank
Is it
you.
clear? So just one last, if there are other questions, feel free to ask. Stop me and ask questions.
सर एक क्वेश्चन था.
जी प्लीज.
सर मुझे कंफ्यूजन हो रही है होमोलोगस के अंदर और सिस्टर क्रोमेटिड्स में ना, कि मतलब होमोलोगस इन चारों को हम मिला के होमोलोगस बोल रहे हैं, या मतलब वो जो दो सिस्टर क्रोमेटिड्स
नहीं, हाँ, इन चारों को भी आप होमोलॉग होता क्या है? होमोलॉग का मतलब है आइडेंटिकल क्रोमोसोम्स. क्रोमोसोम्स एग्जिस्ट इन पेयर्स इन डिप्लॉयड सेल. हमारी बॉडी का हर सेल एक्सेप्ट स्पर्म एंड वो साइट्स वो डिप्लॉयड है. उसमें होमोलॉग क्रोमोसोम्स या एक क्रोमोसोम सिंगल नहीं एग्जिस्ट करता, वो पेयर्स में उसका पेयर होता है आइडेंटिकल. ठीक है. ये जो स्पेशल है ना, ये चार जहां पे सिस्टर क्रोमेटिड्स हैं, ये इन क्रोमोसोम्स को इस फॉर्म में हम कहते हैं माइटोटिक क्रोमोसोम्स. ये नॉर्मली नहीं हमारे, ये सिर्फ तब नजर आते हैं जब सेल डिवाइड कर रहा होता है.
ठीक है
Which
सर.
means DNA replication has taken place, which means all the DNA sequence on this single chromosome वो replicate करके sister chromatid बन गया. Did I answer your question, Salman?
यस सर, समझ आ गया. जज़ाकल्लाह सर.
Anyone else?
Yes, sir. I just want to build on to his question.
Yeah.
ये जो एक चार का सेट है, which is this sort of cross figure, this is one chromosome, और एक सेल में के अंदर there are 46 of these, right?
नहीं, ये जो चार का सेट है, ये एक इंडिपेंडेंट क्रोमोसोम है. ये जो इसका पेयर है, ये इसका होमोलॉग है. तो ये बेसिकली दो क्रोमोसोम्स हैं.
Okay.
नॉर्मली डिप्लॉयड में ये 46 ये होते हैं, ये वाले. मतलब लेट से मैं इधर क्लीन करके ड्रॉ करता हूं. ये क्रोमोसोम 1 आ गया, और हर क्रोमोसोम की अपनी शेप होती है. ये क्रोमोसोम 2 आ गया, इसका होमोलॉग भी हो गया. ये क्रोमोसोम 3 हो गया, इसका होमोलॉग हो गया. इसी तरह आप चलते जाएंगे तो इस तरह आपके पास 23 पेयर्स होंगे, which will make 46 chromosomes. Clear?
Yes.
In a diploid cell. But when cell decides to divide, it has to replicate. All the DNA has to undergo DNA replication through this phenomenon so that chromosome gets duplicated. What makes up a chromosome? It's DNA. DNA gets duplicated. तो जो chromosomes हैं, each chromosome look like this. तो उसका homolog will also look like this. Clear? तो before cell divides, तो उसके पास 46 ये वाले पेयर्स होंगे, ये वाले.
Okay.
How many sister chromatids होंगे?
92.
92. Very good. ठीक है. Which is an indication कि पूरा DNA replicate हो गया.
Okay.
और
तो
ये
सर,
सेल जब
replication-
डिवाइड होगा सेल डिवीजन के थ्रू, इस प्रोसेस के थ्रू एम फेज में से, तो वो 92 जो है वो 46 92 सिस्टर क्रोमेटिड्स विल बी सेपरेटेड, और आपके रीस्टोर होंगे 23 पेयर्स ऑफ होमोलॉग क्रोमोसोम्स इन ईच, व्हिच मींस 46 क्रोमोसोम्स, जो कि
46.
इस तरह होंगे. क्लियर?
Yes, sir.
May I
Thank
answer
you
your
very
question?
much. Yes, sir. Thank you.
Anyone else? अच्छा, एक बड़ा इंपॉर्टेंट पॉइंट है, कॉन्सेप्ट है इसमें, जो आप में से किसी ने भी क्वेश्चन नहीं पूछा. वी हैव 6 बिलियन बेस पेयर्स, एंड इज दिस मशीन डीएनए पॉलीमरेज और डीएनए रेप्लिकेशन मशीन, इज इट सो प्रिसाइज दैट इट डजंट ऐड अ मिसमैच? इज देयर चांस ऑफ एन एरर?
आपने
Chance of error
times
है.
there are mutations.
नहीं, आपने मेंशन किया था अपने लेक्चर में कि देयर इज आई थिंक 1 in a billion चांस होता है कि एरर हो जाए, लेकिन हो भी जाती है तो डीएनए पॉलीमरेज के अंदर एक इनबिल्ट सिस्टम होता है जिससे वो करेक्ट कर देता है. आपने उसको कहा था कि वो प्रूफ रीडिंग कर लेता है कि अगर कोई मिसमैच होता है तो फिर वो करेक्ट कर लेता है उसको.
Excellent. You made me very happy. So you have really looked at things very carefully. So proofreading activity is the one as DNA replication is going on here, as it's moving, there's a scanning mechanism. It scans कि यार मैंने कहीं mismatch तो नहीं कर दिया, because mismatch is going to create an error, and error in DNA replication means mutation. Change in a nucleotide sequence is a mutation. At this moment, we may not realize कि ये error G की जगह वो A ऐड कर गया है, G का कॉम्प्लीमेंट्री. And if this remained unchecked and it goes through DNA replication, here it ends up in a daughter cell. Now this daughter cell divides into two. It will keep passing on this error, and it turned out that this was a very essential gene, which is a tumor suppressor gene. इसमें म्यूटेशन आ गई है और वो कैंसर हो जाएगा, और जनरेशन आफ्टर जनरेशन यही काम चलता जाएगा. So this is a very important concept, proofreading. Any other question you want to ask? Is the way they proved DNA as hereditary molecule, is that clear to you all? That was a beautiful experiment. That's one of the finest experiment in biology.
Sir, I was confused about one point of that.
Hmm?
They grew the bacteriophages in the sulfur radioactive material and the phosphorus radioactive material, right? But like the sulfur. So I'm sorry for the background noises. I'm like
No problem. You go ahead. Go ahead.
outside of. Yeah. So I just wanted to confirm that like जो सल्फर है वो जब उस बैक्टीरियोफेज में ग्रो कर रहा होगा the sulfur because amino acids में जाता है therefore it's wasn't in the, present in the palate and DNA के जो amino acids होते हैं उनके अंदर phosphorus backbone होता है
डीएनए के अमीनो एसिड नहीं होते, डीएनए के न्यूक्लियोटाइड्स होते हैं.
sorry न्यूक्लियोटाइड्स के अंदर जो बैकबोन होती है phosphate की इसलिए वो like pellet के अंदर present था because DNA is injected in the pellet.
So
Okay,
that experiment was, so none of you ask a critical question. इस चीज को critically आपको देखना चाहिए था कि viruses are dead particles. Viruses are not living organisms. So how are you growing viruses in you said कि हमने experiment में viruses grow किए in the presence of radio labeled phosphorus or sulfur, phosphate or sulfur. Is it a valid question I'm asking?
So I guess the bacteria would have been used. I'm not so sure.
Yes, you grow first bacteria and you bring in viruses, and in the media in which you are growing, you have, this bacteria is growing with P32, and one of the bacteria is growing and you are bringing in viruses with S35, radio label S35. And then once the bacteria is killed and you isolate viruses, complete viruses, you isolate viruses. Now, all these viruses which will come out of this isolation mean completely से कोई bacterial cell इसमें आता ही नहीं है साथ, ऐसे ही हम ultra centrifugation वगैरह करते हैं इस तरह. All the proteins of this virus will be S35 and P32, because phosphate is part of the DNA, so the genome of bacteriophage will be P32. तो इसमें आइडिया ये था कि अब हम ये वाले जो हैं वायरसेस, जब हम इनको स्पिन डाउन करते हैं इस पूरे रिएक्शन को, तो हमारी ट्यूब में बिकॉज बैक्टीरिया आर हैवी, बैक्टीरिया सेटल डाउन हो जाते हैं एंड इधर भी बैक्टीरिया सेटल डाउन हो गए क्योंकि दे आर हैवी. और वायरसेस जो हैं वो क्योंकि उनका जो डेंसिटी है दैट इज नॉट राइट वन फॉर द स्पीड जो सेंट्रीफ्यूगेशन की स्पीड हम लोग यूज कर रहे हैं, तो वायरसेस जो हैं वो इधर सुपरनेटेंट में ही रहेंगे. नाउ सिंस वायरस इंजेक्टेड डीएनए एंड वायरस की जो डीएनए सिंथेसिस हुई है, अगर तो P32 है तो जो न्यू डीएनए सिंथेसिस होगी इस बैक्टीरिया के अंदर, द वायरल डीएनए विल एक्वायर फॉस्फेट एंड इफ डीएनए was injected, it will be normal DNA synthesis without any P32 because here we have S35. All the proteins जो virus की नई बनेगी, coat protein, collar, etc., they will contain S35. Now, when we will have बैक्टीरिया उसमें आपके सेटल डाउन हो जाएगा, तो व्हाट वी विल सी कि इधर तो जो बैक्टीरिया है दैट, या जो पैलेट है हमारी जिसमें डीएनए भी सेटल हो गया, दैट कंटेन्स P32, बट क्योंकि बैक्टीरियल पैलेट है ना, तो बैक्टीरिया के अंदर डीएनए गया है तो P32 वो है, लेकिन जो वायरस है, जो कि प्रोटीन्स हैं, वो सेटल नहीं की हैं। एंड व्हाट वी कंक्लूड और इन्फर कि जो बेसिकली वायरस इंजेक्ट कर रहा है मॉलिक्यूल, विच इज द मॉलिक्यूल ऑफ हेरिडिटरी, द कोड फॉर वायरस विच इज रिस्पॉन्सिबल फॉर मेकिंग न्यू वायरसेस, the molecule for inheritance is actually DNA and not the proteins. Clear, Arij? Any
Sir,
other
can I
question?
ask further on this? So
Yeah,
basically,
please go ahead. Please
now,
go ahead.
let's say that once this S35, the sulfur, it finishes, right? It's used up to make proteins. Afterwards, the proteins that are formed, they'll be normal, right? Without the S35, right?
So the
So
question
that's why it's not hereditary.
yeah, so the question I have understood is that once all the S35 is utilized or incorporated in the proteins,
Mm-hmm.
all the new proteins which will be synthesized, they will be without S35. That is the question.
Yes.
If we hypothesize that all the S35 is utilized, then you are absolutely right that all the new proteins will be without S35. But
So that's why we say it's not hereditary.
no. Here my answer.
Okay.
We add ample amount of S35 that, and we do the experiment in a very narrow time window, that S35 is not limiting factor. Clear?
Okay. Mm-hmm.
Now, I think you said something which sounded very dangerous to me. Limiting factor of S35 is not the reason that protein is not hereditary molecule. This whole experiment is actually done to prove whether DNA or protein is the molecule of inheritance, which is responsible for continuity of our life or traits or what we call the hereditary. Now, this experiment proved, because this was much before the discovery of DNA structure. Later on, we discovered that DNA and protein are related. DNA is the code and protein is the decoded DNA. So what we discovered in this experiment is that virus, which is, once it is within a living cell, it is living molecule. Okay. The molecule it is injecting within the living cell, which is bacteria here, is actually DNA, which is then decoded and new proteins are being made. So it means that DNA is the molecule, which is the hereditary molecule, which is passed on from one living organism to its other. Viruses are non-living, but within the cell, they become living. Did I answer your question?
Yes, sir, I got it. Thank you.
Any other question you have? If there are no more questions, I think, is it time is up or you want more time? I am at your disposal.
Sir, I had a small question, if you could
जी हमजा.
answer that. So I wanted to ask, वो आपने slide में proof reading वाले में 3 चीजें लिखी थी, एक तो proof reading, second mismatch repair protein और third excision repair. तो sir इन दो में क्या difference है, mismatch repair protein और excision repair?
Very good. So normally proofreading, good question. Proofreading is when DNA replication is taking place, okay, and you by mistake add a mismatch, the enzyme, the DNA polymerase 3 scans and then removes this base and repairs with right one, okay, and the repaired DNA does not contain any mismatch. Now the other two were base excision and nucleotide excision. Nucleotide excision. Now, DNA molecule, let's say here we have a mismatch. Now, this kind of repair, this is a repair mechanism. This is a proofreading is happening during DNA replication. DNA replication. But outside the cell cycle, let's say I am a living cell. I have my 46 chromosomes. And the person where I am residing in the lungs of a person who is smoker, smoking a lot, or alcohol, or he is taking drugs, anything DNA damaging agent. Now, or I'm exposed to, let's say, some radiation. My DNA gets damaged. In cells in my lungs, they are exposed to some mutagens, and then this mismatch is created. DNA damaged. Now this DNA damage, the repair machine, there's a dedicated repair machine. It tried to repair, and by mistake, originally it was A, it added G there. By mistake, because you have maybe a femtosecond, 10 raised to power minus 12 maybe of a second. To repair this, you cannot take risk as a living system. And by mistake, it added G. Now the system recognizes immediately a red flag is raised that, hey, there is a mismatch. There's a DNA damage repaired, but a mismatch. You have to come in and immediately repair this. Remember, all this is happening beyond DNA replication or cell cycle. Now, there are two mechanisms. One is base excision, the other is nucleotide excision. Base excision is simple one. The repair enzyme will come in and it will recognize, yeah, there is adenine in the parental strand, and this G should be removed. Now, if this G has to be removed, base excision means it just removed the nitrogenous base. We have nucleotide here, phosphate group, the phosphodiester, and here is the guanine, the base. It will come in, it will precisely remove the guanine and replace it with thiamine. A will be repaired with T. The other one is nucleotide excision. Do you know the difference between nucleotide and base, Hamza? What is a
Yes sir,
nucleotide?
न्यूक्लियोटाइड में phosphorus ग्रुप भी होता है और वो शुगर भी होती है और वो भी होता है, लेकिन जो बेस होती है वो सिर्फ नाइट्रोजनस बेस होती है.
So now nucleotide excision, as the word is indicating, in this case it will not be just the base. The repair machinery will come in, okay? It will identify the nucleotide where mismatches. It will excise 4 nucleotides from this side, 4 from this side. A complete chunk will be removed like this, and then the polymerase will add the correct complementary nucleotides. Did I answer your question? So that is nucleotide excision.
Yes
Any
sir,
other
thank you very
question?
much, I got it.
I hope by the end of this lecture, so I wanted to actually make it clear the relationship between, why am I, sorry. Can you still see my screen? Do you see my screen?
Yes, sir.
Yeah. So what was very important in this lecture, because I taught you DNA replication, what I wanted you to be clear about is, which slide should be the most appropriate? This one. I wanted you to learn that DNA replication is not an independent phenomena. DNA, genes, chromosomes are not, something totally different or irrelevant things. They're all connected. But in the context of cell cycle, I wanted you to know why DNA replication is required, where it is taking place, and what is the role of DNA replication in overall scheme of life of a cell. Because cell is dividing mitotically from one cell to the two cells. It cannot complete this cell division if DNA replication is not taking place. And DNA replication, we learned there are leading strand, lagging strand, or continuous or discontinuous strand. There is a whole machinery, DNA replication machinery, in which helicase comes in first, uh, primase comes in, adds a RNA primer, DNA polymerase 3 comes in, followed by DNA polymerase 1, which hydrolyzes the RNA primer, and then ligase completes the phosphodiester bond, and we have, complete DNA replication, and we can see, this sister chromatids. So you must have heard about the mitosis and cell division, but I wanted you to bring the DNA replication in this particular context. Is it clear? Any other question? Do we have time still, or am I running out of time? Oh, I don't know my watch clock is correct or no. My office clock is always running 15 minutes ahead. Abdullah, are you there?
Yes, sir. It's 5:10.
Oh, it means my clock is right today. So thank you very much. Are there other questions? I'm going to upload the next lecture. And I hope today's this exercise, what we did, it made whatever deficiencies were there, the questions you had, they are clear to you. Still, if there are some questions, you can write me an email. You can, if you are on campus, you can catch me anywhere, ask questions, go to your tutorials. So what you have as recitation, today we had the recitation already, it's done. Go to tutorial and make life of your TAs hell with all the questions, any concept which is still unanswered. I'm going to upload after this lecture. I'm going to upload the next lecture. So you'll have another 48 hours for watching video of the next lecture. Any question? If there are no more questions, should I stop it here? I wish you all a good evening then. Thank you very much. Allah Hafiz.